Well, first of all, I would like to thank the organization for inviting me and for giving the possibility of presenting data related with incorporation of paromomycin in liposomes as an alternative strategy for treatment of infectious diseases. Well, according to World Health Organization, more than 25% of the total deaths that occur worldwide are due to infectious or parasitic diseases. In fact, when we talk about parasitic diseases, one a very problematic infection deals with leishmaniasis. In fact, it affects 350 million people worldwide, and it may present different clinical indications that goes from mucocutaneous, cutaneous, that usually is the more typical infection, but visceral leishmaniasis is is the most problematic one, because if non-treated is able to kill the people. In fact, visceral leishmaniasis was responsible for 28,000 deaths in 2012. So, in fact, very few therapies are in nowadays for the treatment of visceral leishmaniasis, and particularly most of them have been introduced 5 decades ago. And so these therapies are associated with severe side effects, and the worst is the appearance each year of development of resistance. So it is urgent to appear new therapies. Of course, the most effective therapy against visceral leishmaniasis is ambisome, the liposomal formulation of amphotericin B. In fact, is the most effective therapy, but the high cost enables the use in large areas that are where visceral leishmaniasis occurs, characterized by usually poor countries. And so, of course, the incorporation of amphotericin in liposomes was able to reduce the nephrotoxicity associated to this molecule. So we think that if other molecules, other therapies appear, we can reduce the price of these liposomal formulations. Actually, I would like to talk about paromomycin, that has been approved in the last decade in the free form for the treatment of visceral leishmaniasis using the intramuscular injection. But even some slightly promising results using this molecule in the free drug, this molecule presents an inappropriate biodistribution profile. And so, based on this, our strategy was to improve the therapeutic performance of this molecule by incorporating in liposomes as a way to improve the accumulation in liver, spleen, so the main infected organs in case of visceral leishmaniasis. Well, when trying to develop one liposomal formulation, we have to fulfill several requirements. Okay, we have to select the best, and that's what we have done when we developed this liposomal formulation of paromomycin. We had to select the best lipid compositions that were able to incorporate in a higher extent the antibiotic. So it means that we have to maximize the amount of the antibiotic per mole of lipid. Also, we had to check if the formulation was stable in suspension, in biological fluids, and most important, we had to be sure that the formulation in lyophilized form, that corresponds to the best way of preserving the formulation, the liposomal formulation, we had to be sure that we were able to preserve the physicochemical properties. And so when each time we needed to use the formulation, we just had to rehydrate and to be sure that the mean size and that the antibiotic was incorporated in liposomes. And this was possible using a cryoprotector, the trialose. Also, when we think about infectious diseases, we have to be sure that the formulation is able to internalize in macrophages, and moreover, that they are able to exert intracellular activity. Well, the rest we have to see when we inject it in mice. We had to see the biodistribution profile of paromomycin that in liposomal form versus the free form, because we know that we knew from literature that paromomycin in the free form is rapidly excreted. And so from here I do not see, but you will see that it appears four columns, each color, and the lowest correspond to the percentage of injected dose per gram of organ that was observed at 2, 6, and population in those organs was very high. So, based on these results, we went to check the therapeutic effect in the murine visceral leishmaniasis model of infection. And in this model, in this first screen, we used a high infection dose. So we injected in mice 10 to the 7 promastigotes intravenous. And 1 week after, we started the treatment for 5 days. And here in this figure, it appears the parasite burden per gram of organ in liver and spleen of different groups of mice. So we see the first 2 columns correspond to infected and non-treated mice. The other 2 correspond to empty liposomes. So we wanted to check if the liposomes themselves were able to exert any effect. And in the 4th group corresponds to paromomycin administered in the free form. And we can see when we compare the free form and the control group, the therapeutic effect is not pretty good. Okay. Then we also used a positive control, glucantine, that is used for treatment of leishmaniasis. And here our liposomal formulation, the 2 last columns. And we can see that the administration of paromomycin using this scheme was the most effective regarding all the treated groups. And taking this into account, we wanted to check if liposomes were effective in early treatment and also in a delayed treatment. And for this, we used a lower infection dose, so we injected mice with 10 to the 6 promastigotes, and we have done started the treatment 1 week after infection, just like in the other. And what we observed is that, again, the control group very similar to free paromomycin, while for the liposomes in the liposomes containing paromomycin, the parasite burden was below detection limits. That even corresponds to around 200 promastigotes per organ. And, okay, this is in early treatment, and in a delayed treatment, we were able to see that paromomycin was still able to reduce the parasite burden. Of course, the infection level was very high. We see that for the control was 10 to the 10, and we were able to reduce 5 logs between the control and the liposomal paromomycin. Taking into account what is described in literature using ambisome, the similar strategy has been done, but in that paper they used a higher therapeutic dose. But here we always use the same, so the 15 milligrams per kilo of body weight. So, taking in this, we wanted to see if 15 milligrams per kilo of body weight was enough to reduce the parasite burden, so we wanted back and check the influence of the administered dose. So they ranged from 1.5 to 15 milligrams per kilo, or on the other side, for free paromomycin, we used only the 15 milligrams because we know that in this model it's not very effective. And according to the obtained results, we could see that for doses ranging from 3 to 15 milligrams per kilo, we were able to clear, at least to below detection limit, in liver the parasite burden. In liver. In spleen, the infection, the reduction was not as easy. But we wanted to compare, okay, we have compared with glucantim, we wanted to compare with another liposomal formulation, and we checked when we compared the effect of paromomycin liposomes versus ambisome. And for this, for the free paromomycin, we used only the highest dose. For liposomal paromomycin, whole doses, and for ambisome, 1.5 and 3 milligrams per kilo. And what we see here is that what we observe is the reduction in percentage of the parasite burden. And using free paromomycin, this reduction is below 60%, either in spleen or in liver. On the other hand, very comparative results were achieved for ambisome or for liposomal paromomycin. And so, conclusions, okay, we can say that we were able to incorporate very well, it was stable, the biodistribution of liposomes was pretty much higher in comparison with free, and we think that the comparative in vivo evaluation of paromomycin liposomes and ambisome suggests that these paromomycin liposomal formulations may be an alternative to ambisome. I would like to say the people that collaborate in these results, the Faculty of Pharmacy from the Universidade de Lisboa, the Universidade do Porto, and I would like to thank Gilead for providing the ambisome. And thank you very much for your attention.
Thank you very much. Also, you managed to do
in time. Okay.
You speed up anyway. We have time for a couple of questions. Yeah?
Hi. Thank you for this presentation.
Okay.
Maybe we'll talk later about a few aspects of this loading, but I was wondering if you were ever if you would consider, because paromomycin is a weak base. It has five amino groups, so it could be easily used for remote loading into liposomes.
Well, in this I have worked with other molecules. One which is rifabutin, and I was able to incorporate using the
Remote
the
loading.
remote loading. In this case, paromomycin is very hydrophilic molecule, and so we use the rehydration method that is very it allows very high encapsulation parameters because we put the lipid with the
Paromomycin. You mix them.
paromomycin,
Yeah.
we lyophilize, and then we rehydrate in more than one step in order to maximize the encapsulation. Actually, the possibility of remote loading, I think it is because I have already worked with Dr. Medicine using the remote loading. In this case, I think it won't be necessary to use the remote loading, and I think it is more easy and even more easy to scale up and to go further. Of course, remote loading, we always can have the liposomes, the empty liposomes, and then when it is necessary, it's just to mix and so on. But we were able, using lyophilizing in the presence of cryoprotector, to preserve the properties of the liposomes, just how they were before.
Okay. Thank you. Thank you.
Any other question, please? Okay, do you have any plans for other kind of in vivo testings, let's say,
Well,
with dogs, for example?
A dog, so it's in another model.
Oh,
It's
yeah,
in
yeah,
cancer.
I know.
Because I work in different areas. Okay, my research area is drug delivery, okay, with particular liposomes, but using different molecules, and according to the molecules, the different in vivo. Where I have worked with tuberculosis, AVIL, and also now with melanoma, but with new molecules synthesized by the university where I now belong. But
Okay.
okay.
Thank you very much. Okay, here, just to present you with the certificate.
Okay, thank
Very
you
good.
very much.
Congratulations.
Okay, thank you. Okay, I know that you are very fond about leishmania.
Yeah. Actually, because we have a project on the vaccination.
Okay,
We
well,
do vaccination on leishmaniasis.
but until now there is nothing.
We have some regulation.
Yeah.
Okay.
Okay.
Cancer, okay.